if you want to learn how to use CUT and PyMol. So that's all on that file, on that drive. So once he copied that, share with you, you should be able to see all of them. So I guess we'll do more CUT and PyMol tomorrow. So please check the primers. So there's a folder called PyMol, a folder called CUT. So that's what we'll do tomorrow afternoon, the session afternoon tomorrow. So look at the primer and then get a feeling as to how to, you saw some of that yesterday? Yesterday, right, yeah. So that's explained in the primer, how it's done, okay? And then we'll go through that tomorrow. Okay, so next part, I'll give you some general introduction to protein structures. How many are familiar with protein structures? Whoa, okay, good. So I'll skip over this slide, right? Thank you. Skip over this slide too. Ramachandran plot, you're all familiar with. And which one's alpha helical, which one's beta? Go ahead, if you know the answer, say something. So which region is alpha helical, which region is beta? This is alpha helical right here? Yeah? That's beta? I'm sorry? Okay, beta sheet, is that what you said? Okay. All right, so beta up here, alpha helical down here, and then over here is really not really possible because essentially stereo hindrance we talked about. And I'll skip over this, definition torsion angles. We don't need to worry about that too much. So the basic concept here essentially, suppose this concept you probably know as well. A protein of 300 residues, and then each residue can have 3 conformations, alpha, beta, or loop. And then based on very simple-minded consideration, you will say the total number of possible structures will be 3 to the 300 power, right? If each one is independent. They're not completely independent, but anyway, bear with me. So that's a huge number. So the vast majority of that is not possible, but even if like 0.0001% that is possible, you still talk about a huge number. So then the idea is actually we're talking about is how, well, one thing is how does a protein, if a protein has 300 residues has so many different possible conformations, how does it ever find the correct conformation? So that's something called the Leventhal's paradox. He was the first one to realize this problem. Of course, in reality, this is not a random search process. It's a multi-directed process how protein folds, but that's beyond what we want to talk about. The idea we have is protein fold here is a unique organization of secondary structural elements in your protein. Okay, how these alpha helices and beta strands arranged in the structure. That's basically what I just said. How they are arranged, how they are connected. So the emphasis is more on the topology, how they are arranged, rather than the detailed exact positions of some beta strands or alpha helices. So you'll see some examples where two putting on the same fold, but some have additional decorations. And they are allowed as well. They don't have to be exactly the same. So this we talked about yesterday already. So this is where you find protein structures. So hydrophobic core, you all know that concept? Yes? All right. So proteins are functioning in aqueous environment. The hydrophobic side chains or the hydrophobic are buried in the core of this protein or the charged side chain on the surface. So then that's the hydrophobic core. Throw away the top half, then it's almost all yellow here. That's the hydrophobic core. So one game we'll be playing today is where in the world is the hydrophobic core? You know the game? Or where in the world is Carmen Sandiego? Okay, you don't know, that's fine. That's a US game. Anyway, the other thing is when you look at a protein structure, they are very complex. So here is a protein of myoglobin again, 153 residues. So typically, each residue of a protein contains about 8 non-hydrogen atoms. That's what's being shown here. So essentially, 8 times 153 is somewhere around 1,200 atoms. So it's a very complex structure. That's why you don't really see, you see a forest, you don't really see information. It's too much information, I guess. You're swamped by the information. So that's why you'll see this. So I just said that already. So they're generally schematized. So which you're familiar with. Alpha helices as these helical ribbons, and then beta strands as arrowed ribbons. Okay, so that's what you'll see today. Of course, in detail, the detailed region on the C are still the ones you can show as in all atom models. That's something we'll look at tomorrow, Thursday, and Friday. Today, we're zooming out, looking at only the overall molecules, how the fold looks like. So that's basically, you're looking at schematized structures like these. Okay, so next, so that's the introduction part. So now we're going to go through, look at some example proteins, and mention function a little bit along the way, but mostly just look at how proteins folds look like. Some famous, if you will, more popular folds. So the protein structures, the folds can be divided roughly this way. There might be different ways to divide them. You can say all alpha proteins. That means the structure contains all alpha helices. But you could have one or two small beta sheet, one small beta sheet or two couple small beta strands, okay? All beta proteins, I can imagine all beta strands or beta sheets. But you could have one or two helices in there as well. That's okay, you'll see examples of that. And then these two are different. I'll explain that along the way, and then others will come to. So here's the alpha helix. I guess you're all very familiar with that. Okay, side chains we point out. Yes, here the side chains all point down. That means that this is the N terminus, this is the C terminus. All right. So now start looking at a few structures. The simplest all helical structure will be something called the coil. So two helices winding around each other as a coil. Okay, so that's a very common structure as well. So the unique unit is about 140 angstrom or so. So that's found in nature. Your hair is basically something like that. And then if you look at coil structure, oftentimes I'm doing as a sequence, so the coil assumes some kind of a unique signature. And that's due to the fact that the two helices need to wind around each other. So again, where in the world is hydrophobic core? The hydrophobic core is where the two helices touch each other. Okay? So if you look at these two helices, this is from the side, this is from the top. So you start from residue A right here. Okay? And then next residue B, C, D. Okay, that's one turn. One turn of helix is 3.5 residues. So essentially, you end up here. 3.6 residues per turn. So you end up next to A, but not exactly on top of A. So you go one more round, E, F, G, and then H. The next one will be 7.2. 3.6 per turn, 2 turns will be 7.2. So residue D will be almost exactly on top of residue A. Does that make sense? Okay. So essentially A and H will be on top of each other. And so that means if you look from the side, this is A, this is D, which is right here. Next one, H, will be on top of that again. So that's basically going around. So it has a periodicity of 7. Does it make sense? So if you look at the primary sequence of the protein, they have something called the heptad repeats. Repeats every 7 residues. And if you look at it, A and D are facing each other between two helices. So they will tend to be hydrophobic. So if you look at the sequence then, position D is almost, in this case, all leucine. A has some variability, but also mostly hydrophobic as well. So that residues A and D that form the hydrophobic core. That's why they have this pattern. That's so-called heptide repeats, which means for these repeats, typically A and D positions are hydrophobic that allow the formation of this coil. And then, of course, that's the hydrophobic core. And on the surface, you can have ionic or hydrogen bonding interaction. In this case, unpairs between lysine and glutamate and this and that. So that's the other positions of the helix. So this is one way of recognizing whether your protein can form this kind of coil structure, whether you have heptide repeats. If your protein contains heptide repeats, chances are you can form this kind of coil structure. It may not be very, very long, could be a local area, but still, you can have this kind of structure. Okay? So this is something you'll probably read about as well. And then another example, very commonly found for alpha helical structures, something called a 4 helical bundle. 4 helices arranged together, forming a bundle together. And then obviously, in this case, the hydrophobic core will be in the center of the 4 helices. Okay? But then you have 4 helices. There are many, many ways of connecting them. So the simplest one, for example, is cytochrome B. So the 4 helices are up and down. So here, red is the N-terminus. So at the N-terminus, you go up in the helix in red. Then you come right back down in green, in the green helix. Then you go right back up in the yellow helix. Then you come right back down in the blue helix. So then if you draw as a topology, up helix, down helix, up helix, down helix, up, down. Okay? Simplest way of connecting 4 helices. And that's a cytochrome. But then, as I said, there are many, many different ways. Another way, very important to us, is human growth hormone. And that you can see red again, N-terminus, up a helix, and then it crosses over all the way from the top to the bottom again. And then up a helix again in red, and then come right back down in the blue helix, but then it crosses over to the top again, and then come back down in the 4th helix with the C terminus. So then in this one, you have up, crossover, up again, and then down, crossover, down again. So that's human growth hormone. Why did we do it that way? Don't know. So that's what we see. Okay? So this is somewhat more complicated, but then many other ways of connecting them have been observed as well. Okay, so 4 helical bundle. And then a third very common structure for all helical structures, so-called the globin fold, because they're found in myoglobin and hemoglobin. And this fold contains 8 helices. They are named alpha A to alpha H, or A through H, alphabetically, essentially. So 8 helices, if you count it out. And of course, the role of the globin is by heme, which is an oxygen carrier. So myoglobin is a monomer, hemoglobin is a tetramer. So there are 4 chains of the globin fold. Each one of these colored chain is a single molecule, and that molecule has the same fold as myoglobin. Okay? We'll come back to hemoglobin a little bit later as well. So I have some movies with this as well. You see the 4 helices, and you see the heme. So with this kind of schematic or schematized representation, you can clearly see the biochemistry that's important for this molecule. That is essentially, there's a heme right here, and that's oxygen right there. So oxygen binds to the iron in the middle of the heme, and the heme is bound through the iron to a histidine side chain, which I show right here. So then at the other side, the 6th coordination position of the iron is occupied by the oxygen. Okay? All right, that's what I'll show you about all alpha structures. Now let's talk a little bit about all beta structures. If you have questions, please just raise your hand and then let me know. If I'm going too fast, let me know as well. So far, okay? Too slow? No No response. Okay, that means I'm okay. Okay, beta strand. I guess in case so if you look at a beta strand structure, so here's a beta strand, it's pleated because this one is amide plane, another amide plane, another amide plane, another amide plane, right? And the side chain, if you look from this side view, side chain pop in opposite directions. One left, one right, one left, one right. So then if you look from the top, then the main chain goes back and forth. The CO goes this way, this one goes that way, this goes this way, this goes that way. So they're flip-flopping. So the amide plane is flipping 180 degrees as you go along the chain. So that's why the side chain is also popping off. Okay, so beta structures, a single beta strand, I showed you already, but that's not going to be happy because all the hydrogen bonding, they don't have hydrogen bonding partners. In the helix, you wind around, the hydrogen bonds are made within the helix, right? So that's happy. A single beta strand by its own, it doesn't make any hydrogen bonds, right? So here, hydrogen bond donor, acceptor, and donor, acceptor, and over here, same thing, donor, acceptor. So these can all make hydrogen bonds on the main chain, but a single beta strand does not make any of them. So it's not going to be a happy situation. So we're not going to talk about single beta strand. So we have to talk about beta sheet. Beta strand coming together forming a sheet-like structure, but not like a sheet in the sense of a perfect sheet of paper, not flat. And they're actually twisted quite a bit. I showed you that example yesterday. A 6-strand sheet twisted 90 degrees. So you have a beta sheet, and then you can have a beta sheet twisted so much that it folded on its own. Okay, so that's a beta, well, sorry, getting ahead of myself, too hungry. So beta sheet, a single sheet, which we'll not talk too much about, that's boring. Well, sorry, not boring. That's something that's simpler. And then you can have two sheets on top of each other, you get a sandwich. Okay, you can have sheets fold back on itself so the beginning and end touch each other. You get a barrel, something to drink with a sandwich. Okay, so I'll show you some examples of beta sandwiches and beta barrels, and then actually something else I'll show you a little bit even more interesting, beautiful I should say, that you can form a beta structure. And then a single sheet, all beta structures now stable in the sense that a single sheet will not have a beta hydrophobic core. This is all considering the idea of hydrophobic core. Without a hydrophobic core, it will be less stable. So by the same token, a single layer structure would not be stable. Okay, now let's talk about beta sandwiches, which are made of beta sheets. So when you look at sandwiches, you find sheets as well. So a very popular and important beta sandwich is something called the immunoglobulin fold. That's present in your antibodies, immunoglobulins. So immunoglobulins, antibodies have two constant and variable domains. They have the same fold, but you can see the variable domain has two extra beta strands. But let's start from the constant domain. You can see the structure is a beta sandwich with 4 strands blue in the back, 3 strands green in the front. Okay, and where is the hydrophobic core? Between the sheets. And in this case, there's a disulfide that's conserved among the antibodies that provides additional stability for this beta sandwich structure. So not only you have a hydrophobic core that stabilizes structure, you also have a covalent bond between the 2 sheets. So that's the constant domain. Variable domain has the same fold, even though it has 2 extra beta strands. So again, fold is how things are connected. Even though this structure has unique features, but still, it's a IG fold, immunoglobulin fold. So the 2 extra strands are in the front, C prime and C double prime. So the front sheet now have 5 strands. Okay, but more importantly, the 3 loops on this end of this fold are hypervariable. As they are so-called CDRs, complementarity determinant regions. These are hypervariable, and that's how your antibody recognize different antigens by basically changing the sequences of these loops. You can recognize very different antigens. Okay, so very important structure. So this you could say is the scaffold that help you to present these two loops, three loops that recognize different antigens. That's important for your immunity. For example, well, a little bit behind the times now, SARS-CoV, the spike protein on the surface bound to an antibody right here. So the receptor binding domain right here in this yellowish color, and this is the antibody CDR will be down here. So that's how the antibody works, by neutralizing the spike protein. Okay? All right, so when you think about a beta structure, so you saw a helix, earlier one saw all helical structure, helix go up, come back down again. Same is true for beta structure. You have beta go up, but you can't keep on going forever because then you have a single beta strand. You have to come back somehow, right? You have to change direction. So the simpler way to change direction, not the simplest way, the fastest way of changing direction is over 4 residues. So residue I right here, so CFICO, so this is the residue I, and CFICO residue I plus 1, and CFICO I plus 2, and CFI I plus 3. So here the chain is going up, and then by 4 residues later, it's going down already. Change the direction. So that's called beta hairpin or beta reverse turn, depending on which book you read. You heard about this? Okay, yes, good. Oh no, yes or no? So the nodding head means no, right? I'm not sure. Anyway, so there are 2 ways of doing this beta hairpin. Type 1 and type 2. Can you see a difference between them?
R2 and R3 has different orientation.
The R2 and R3?
Has different orientation.
Uh-huh. Okay, good point. And that's because of what? There's something else that's even more dramatically different. So do you sort of see depth queuing? What I mean is the atom that's in front is obscuring the atom in the back. So the atom in front, you see the whole thing. The atom in the back, you see only a part of it. So that's a depth queuing, right? It'll give you a sense which is in front, which is in the back. Okay, so given that, do you see a difference now? Okay, all right. So this carbon right here. Okay, sorry, I guess I'm belaboring the obvious. Anyway, this carbon here is in front of the oxygen. Where down here, the carbon is behind the oxygen. So essentially, this is the amide bond right here, right? It's a plane. So the plane is perpendicular to the screen here. So this is the carbonyl. All right, let me stand over here. Here, oxygen in the front, bottom, right? And then flip 180 degrees, you have that. See the difference? All right, good. Okay, so that's type 1 with the oxygen pointing to the back. Type 2, oxygen pointing to the front. So now if you look at it, type 2 with the oxygen pointing to the front, R3 also pointing to the front. So would that be a happy situation? Stereohindrance, right? The side chain and the carbonyl oxygen could be clashed or will be facing each other, will not be very happy. However, if you have a glycine here, what happens? Now you don't have a side chain. So then you can assume this kind of conformation. And that's what I showed right here in this movie. So you have here the carbonyl pointing up, just like here. And here you have a glycine, no side chains. Here are the black are the side chains. There is no side chain. So type 2 beta reversed or beta hairpins typically has a glycine at the third position because it's their stereohindrance problem. Glycine, having no side chain, has many, many unique roles in protein structures. It can fit in tight spots where no other residue can fit. That's why it's oftentimes how glycine is conserved, because you need a tight spot so that the glycine can make this turn or can fit in the tight spot. Okay? So this is sort of a simple way. When that stopped. This is a way to make a 180-degree change in direction of the beta strand. Okay, so we talk about human growth hormone. So the human growth hormone receptor also has Ig-like fold. So essentially, the receptor is a dimer, one molecule on the left, one molecule on the right, and a single molecule, the growth hormone, bound between a dimer. So it's a 1-to-2 stoichiometry. Okay, so this is done using by crystallography many years ago now. So each one of these domains is Ig-like, immunoglobulin-like. Okay, so that's beta sandwich. One example I showed you. Now let's talk about beta barrels. So I mentioned up and down, changing direction. So here, the simplest beta barrel will be the up-down beta barrel. So start from the N-terminus, which is over here. Some stuff going on in the back. You cannot see, but doesn't matter. It comes around here, strand 1. Okay? Strand 1 goes down, and then come right back up, strand 2. And then go right back down, strand 3, back up 4, down 5, up 6, down 7, up 8. But you can see 8 right next to 1. So 8 and 1 are hydrogen bonded to each other. So there is no beginning, no end here. Everything is bonded together. So that's the concept of beta barrels. So this is 8-stranded up-down beta barrel. Okay? So now where's hydrophobic core? Where the water is. Or I guess I'll say water, you probably say beer, but whatever. So in the center of the barrel. And in this case, it turns out the barrel center has a little cavity right there. So you have these all hydrophobic residues, especially lots of phenylalanine, methionine, tyrosine. So these are all hydrophobic, but there's a cavity. And the cavity binds retinol. As you see in the movie, retinol is almost all well, not almost all. Yeah. They're all hydrophobic except the very tip of it, which is a hydroxyl. That's why retinol is alcohol. So hydroxyl is left outside, everything is buried. So essentially, it's a binding protein, it's a carrier for retinol. This very hydrophobic molecule. You make this nice little structure to bind retinol. So this is hydroxyl right here. Okay, so that's shown right here. Oh, first topology. So up-down beta barrel, you cut right here, spread it out, up, down, 8 times. Okay, you see some more this kind of topology drawing today as well to help you understand fold. So here's the movie. So side rotation, top rotation. So here's the barrel. Here sideways, you can see retinol, hydrophobic part bound in the middle of the beta barrel, hydrophobic being part of the hydrophobic core now, and then the hydroxyl is the one that's left out in the water, where it's happy. Okay, so the structure makes perfect sense for the function of the protein. And then also you have a helix here, even though we say it's all beta structure, but there is a helix, which is allowed. That's okay. All right. So another example, I'll show you something called the Greek key motif. It's Greek key motif because it looks like the motif that's found in ancient Greek vases. So very simple structure as well. So you have a strand coming down, a flexible loop in the middle, another strand, and then a quick reverse turn. Could be a beta hairpin, I just showed you. And then you have, so 2 and 3 form a 2-stranded beta sheet, and then a loop, and then 1 and 4 form a beta sheet. So very simple structure can be folded using very local sequences. And then what you do here is, imagine you grab the top, and then you grab the bottom, you fold it this way. You will get that. right? So very nice 4-stranded anti-parallel beta sheet. So that's the Greek key motif. It's found in many, many structures, very common folding motif because it's so easy to form. So one of them is chymotrypsin, an enzyme we'll spend some time talk about when I talk to you about proteases. So chymotrypsin contains 2 domains. Each domain contains a Greek key motif. In addition, the Greek key motif is right here, 1, 2, 3, and 4. And then it has 2 additional beta strands. So it's a 6-stranded antiparallel beta sheet that folds onto itself to form a 6-stranded antiparallel beta barrel. So now it's called a Greek key beta barrel because it's a beta barrel that contains the Greek key in there as well. And then the 2 domains have the same fold. Okay, so domain 1, domain 2, each one is a beta barrel. It's sort of seen shown right here. Okay, 1, 2, 3, 4 is the Greek key motif, and then 5, 6 completes the barrel. And then you'll see later, as often case with biochemistry, reactions happen, the active site is located at the domain-domain interface. So serine 195 is right in the interface between the 2 domains. Are you familiar with chymotrypsin? No, that's fine. We'll talk about it. So here is the movie of that. So here's the barrel right here. You can see the barrel. Here's the other barrel. And then, again, a couple of helices, that's allowed. And then serine 195 right here, histidine 57, aspartate 102. So that's a chelated triad if you don't, if you remember. So that's right the domain-domain interface. And then there's another piece in blue, another piece in green that are very important for chymotrypsin as well, which we'll talk about later as well. Okay? All right. So we can go one step beyond that. So I showed you earlier 4 strands forming 2-strand beta sheets. You can have 8 strands. 1, 2, 3, 4 going up, 5, 6, 7, 8 coming down. And you can have these 2-stranded beta sheet. And then you wrap them around a barrel like this. That's called a jelly roll. I'm not a baker, so I don't know what a jelly roll looks like, but supposedly it's how you make a jelly roll. Anyway, so that's how you form this essentially 8-stranded beta barrel by wrapping around the barrel like that. And you can sort of see the 8-stranded barrel actually contains 2 Greek key motifs in there anyway, because the way it's formed, it's just like how we make Greek key motifs. So you have you have a Greek key motif here, another Greek key motif here. Okay? And this general structure is found very often in capsid proteins for viruses. So viruses have a shell, a protein shell that encloses the nucleic acid genome. And that shell made all protein. And that protein oftentimes have this fold. Especially like rhinovirus, the common cold virus, has 3 proteins right here. They all have the same fold. I'll show you an example of rhinovirus later as well. Okay. So you already saw in the case of chymotrypsin, many proteins contain more than 1 domains. A domain is a stable unit of structure, if you will, or separate folding unit. Typically about a couple hundred residues large, but it can be as large as 800 residues. So structures are known, or proteins has 1 domain, but it's 800 residues long. But typically somewhere around a couple hundred. And I already mentioned this, many interesting biochemistry occurs at the interface between domains, just like what we see with chymotrypsin. And this is another example, domain C, domain B, 2 domains. And here's the active site. That's where the catalysis happens between 2 domains. And the domain actually does this, like clamshell, open and close, which happens all the time. You hear about open and close quite a bit this week, if you haven't heard already. Okay, so that's one example of our beta structure. Well, that was the second example. Third example, something called the beta propeller. Okay? And this is also a very popular fold in nature. And this structure specifically comes from G protein. the beta subunit of G proteins. We'll talk about G proteins on Friday, so you'll hear about this a little bit more. Again, the structure starts with the helix. That's okay. They come all the way down, first strand right here. We'll ignore that for now. And then this strand comes over to the center. You have another strand coming up towards you, a strand going down, a strand coming up towards you, a strand going down again. So 4-stranded anti-parallel beta sheet, very simple structure. The trick is this then go to the next one, make another 4-stranded anti-parallel beta sheet, and these two are stacked onto each other. Okay, and then they are arranged all around the circle like this, like a propeller of a plane. And the propellers are made each with a beta sheet. That's why it's called a beta propeller. And interestingly, the sequences of these proteins have a unique pattern. And that pattern is called the WD-40 repeats. And I guess here you probably don't know WD-40. Do you know WD-40 here? No. Okay. So again, some Western kind of a brouhaha, whatever. So W is tryptophan in 1-letter code. If you know your amino acid, what happens is tryptophan is repeated every 40 residues or so. And that tryptophan sits right here. So the reason you have to repeat because each one's 4-stranded beta sheet, so the separation between neighboring sheet is about 40 residues. So you have the tryptophan sitting here, a tryptophan sitting there, a tryptophan there, there. So that's why you have this repeat. It's a consensus repeat, not exact repeat. So it's oftentimes tryptophan, but doesn't have to be tryptophan all the time. Okay. And another very important beta propeller is the neuraminidase of influenza virus. So that's has, go back here. So G beta has 7 propellers. 1, 2, 3, 4, 5, 6, 7 around the ring. Okay? Influenza neuraminidase has 6 propellers. So you can go from 5 to 8 maybe propellers, how many there are. And this enzyme is critical for the life cycle of the virus. So there are small molecule drugs developed against it. Some of you may know Tamiflu, bird flu back in 2000, early 2000s. So I have some in my medicine cabinet as well. So Tamiflu essentially is developed to target this enzyme. It binds the anti-site, inhibits the enzyme. That's how it kills the viral life cycle. Okay, last all beta structure, something called beta helix, as I mentioned. So you know about alpha helices, which are formed by residues, amino acid residues. Beta helices are formed by beta strands. So you can have beta strands arranged in a helical pattern. And then you have beta helix. So for example, this one, a green strand forming a green sheet right here, and the red strand in the back forming a red sheet, and then they are connected using this helical pattern. And then this one has 3 sheets, yellow, green, and blue. And that's shown right here. So first sheet, second sheet coming to the center, and then third sheet coming around right here. Okay, and then again, couple extra helices, that's okay. And the hydrophobic core in the middle of the helix. Okay, so that's found quite often in nature as well. Very remarkable structure to me. Okay, so that's all I'm going to say about all beta structures. These are just examples. I'm not saying all the structures look like this, okay? Just examples, more commonly found. And then let's talk about alpha/beta proteins. Alpha/beta means the alpha, beta, following a pattern. So alpha followed by beta, followed by alpha, followed by beta. In comparison, alpha plus beta means there's alpha, there's beta, not in any specific pattern. So this will cover almost all the proteins, if you will. So I'll show you a couple examples of alpha/beta proteins, important ones. And the most popular protein fold is something called the TIM barrel. TIM means triose phosphate isomerase, which is an enzyme in the glycolysis pathway. if you remember your biochemistry, but it doesn't really matter. So the structure is very simple. It has an 8-stranded, fully parallel beta barrel in the middle. So you can see right here, 1 is here, 2, 3, 4, 5, 6, 7, 8. Okay? Oh, sorry, 1 is here, 1, 2, 3, 4, 5, 6, 7, 8. Okay? So they all point towards you. So all the strands point towards you. So that means they need to go back down again to connect to the next strand. And they go back down as helices. So strand come up, a strand, down helix, up strand, down helix, 8 times. That's what's here. Up strand, down helix, up strand, down helix. 8 times. And then the last strand, number 8, is hydrogen bonded to the first strand, as you see right here. So you have a fully parallel beta barrel. And then, so in this case, this is a TIM itself, this enzyme itself. And then the substrate is bound to the top, where all the beta strands point towards. So you can sort of imagine this structure is polar, asymmetrical, because the top is where all the beta strands point towards, and the bottom is where the intermini of all the beta strands are. So the two sides are different. You can see now in the TIM itself, the substrate binds to the top of the structure near the C-terminal end of the beta strands. As it turns out, actually, that's true for almost all TIM barrels. They all use the top of the structure to form the active site. So as I mentioned, TIM barrel is one of the most common fold in protein structures. So even though you don't see any sequence signature that could be a TIM barrel, but when you look at the structure, actually it's TIM barrel. So essentially, a single fold can support many, many different biochemical functions. So that's the idea of a scaffold. One fold can support different functions. The way to think about it, you can sort of say is essentially by changing the residues around the top of this beta barrel, you can change the biochemistry. A few different side chains here, you can change which substrate recognizes, also change what kind of chemistry it does. Because the fold is so stable, you can mess around with the top of the structure without changing the overall structure of the enzyme. So that's why I think it's very popularly found in nature. Second example of alpha/beta protein is something called the Rossmann fold. And here, rather than a beta barrel, we have a beta sheet in the middle, 6-stranded, fully parallel beta sheet. So in this case, they all point to the top. 1, and then you need to come back down again as a helix. 2, down again as a helix. 3, going up. And then it crosses over. So now 4 is next to 1, 4, 5, and 6. Okay, so if you look at the topology drawing, 1 up strand, down helix, up strand, down helix, up strand, crossover, up strand, down helix. Okay, so this fold is very important for binding dinucleotides, NAD, NADP. So if you talk about metabolic enzymes, they often use NAD and NADP as cofactors, and many of those enzymes contain this fold. And this fold also can bind divalent cation like magnesium. And that's also very important for other cellular functions. Just like TIM barrel, the top of this beta sheet is where things bind. So that you see right here. So top beta sheet, NAD is bound to the top of it. And then you have 1, 2, 3, 4, 5, 6. You have 7 strands here. So that's an extra strand at the end. That's okay. Still a Rossmann fold, even though now it's 7-stranded rather than 6-stranded. That's fine. The general topology is the same. So this is still another example of a Rossmann fold. Okay. So I guess I can say more, but I won't. Okay, the last example I'll show you for alpha/beta proteins is something called the ribonuclease. This is the structure of ribonuclease inhibitor. And very simple structure as well. 17-stranded, fully parallel beta sheet. And then, so they're all pointing up. And then and then you go back down as a helix. So up strand, down helix 17 times. And then this protein is also known as leucine-rich repeats because it contains many leucine residues. So oftentimes, protein contain leucine-rich repeats from this kind of structure. And actually, it doesn't have to be a strand coming up. It could be a helix coming up. So helix up, helix down, helix up, helix down. That's possible as well. Okay. So this may not be beautiful. This is the actual structure. So to me, it's more beautiful than the schematic drawing. So there's a horseshoe-like structure. So where will be the hydrophobic pore for this one?
Between the chain and helix.
Great, right here. And that's where the leucines are. It's leucine repeat. Leucine is very hydrophobic, and that's sitting between the strands and the sheets. Or if you have a helix here as well, then it's between the two helices. Doesn't really matter. Between the two layers, what the leucines are. They are rich because they are needed to stabilize this overall structure. And then you wonder, why did it leave the gap right here? Why can't this strand be hydrobound to that strand make a barrel rather than a sheet? Well, when you know the answer, let me know, please. So this is a ribonuclease inhibitor. So the ribonuclease binds to the center of this horseshoe and somehow that inhibits this activity. So very popular fold as well that you find in nature. Okay, so that's examples of alpha/beta. Alpha plus beta, as I mentioned, cover many different proteins. So I will not show you any of those. I will, well, not today, but obviously you'll see a lot of them in the next few days. And then additional factors stabilize protein structures. Disulfide bonds, you saw the one example already. Metal ions, binding of metal ions, cofactors, and also nucleic acids or nucleotides can stabilize your protein. And in addition, of course, protein can be modified tremendously by PTMs, post-translational modifications. This knowledge is a partial list with modern technology, mass spec, and everything else. You find all kinds of modifications now, lysines, arginines, everything else. So the list can probably another slide probably will not cover everything still. But anyway, you have the idea. And we will talk about phosphorylation quite a bit, protein kinases and stuff. And disulfide, I guess you've seen that. And I came from Columbia, so I had to mention GIP because Marty Chaffee is my department. So you all know about GIP, right? So this is how it works in case you don't remember. So essentially the oxidation of this tyrosine is forming this extended ring structure that allows it to fluoresce. So it's a PTM, post-translational modification. All right? Oh, okay. All right, so that's tertiary structure. And then we'll talk about, we'll have a few more minutes. We'll talk about quaternary structure, how protein molecules come together forming complexes. So this could be homoligomers, same protein forming dimers, trimers, and then 60mers, which are viruses, 60 copies of a protein coming together forming a virus. Or it could also be heterocomplexes, different proteins coming together forming a complex. Either way, just like domain-domain interface, protein-protein interface oftentimes is where important biochemistry happens. So it's very important to forming these kind of complexes. So I'll just show you a few examples. So first is linear oligomers. For example, in this case, tobacco mosaic virus is a virus. So it has in this case, RNA genome, shown right here. And it's coated by its coat protein, if you will. It surrounds this genome. And then if you look from the side, in the EM image, it's like a rod-like structure. Okay. So you can imagine each one of those is a protein. So it's a very large quaternary structure containing both RNA and protein. And then another example of oligomers, not linear but globular, spherical or shape. You can have two molecules come together forming a dimer and trimer, tetramer, hexamer, octamer, cubic structure, and the icosahedral is what you use to form a virus. Most viral capsids have icosahedral symmetry, so it has basically 60 copies of a single protein that come together, forming something that looks like a soccer ball. Actually, in reality, it's more complicated than that. I'll just show you soon. So here, example of a dimer. So one molecule in red, one molecule in yellow. You can sort of turn 180 degrees, and the two molecules be identical. Okay. And then here, the structure of hemoglobin again. So as I said, a tetramer, two different kinds of subunits, alpha subunit and beta subunit, alpha 2, beta 2. Okay. So essentially, 4 molecules, each one binds a heme, as you have here. Okay, and then this is the viral capsid, 60 copies of VP1, VP2, VP3. So 3 proteins, VP1 in green, VP2 in blue, VP3 in purple or magenta, forming this. And then this is repeated 60 times. Either this view or this view to form this capsid. The role of the capsid to encapsulate the, in this case, RNA genome, the genome which will be sitting in the middle of this capsule to essentially keep the genome safe during its life cycle. So that's the whole idea. Okay, so that's the slide I will show, but I will show you something, show you for the virus. So here the structure of the rhinovirus. And so what you have, let me color by chain. So here you have essentially the 4 subunits, the 3 subunits shown right here. That's a unique portion of it, so-called the protomer. But then there are 60 copies of this because of icosahedral. There are 60 copies. So if you click on play, you'll see all the 60 copies all over the place because that's related by the symmetry of this icosahedron. But that's probably, sorry, I don't want this. So let's show all together. That'll put you more. How does that look? Better? So that's essentially all 60 copies of each protein. So total 180 protein molecules forming this capsid. Actually, there's another subunit which is not visible from the outside. It's on the inside. So there's 60 copies of that as well. So there's 240 copies of proteins forming this capsid. So right here, you're looking down the threefold axis. Do you know about symmetry a little bit? Okay, well, we'll learn by fire. So look at this one. So here you have 1, 2, 3, 4, 5, right? 5-fold, look like a star, right? So 5-fold symmetry. And then here you have 3 magenta, 3 cyan. So that's 3-fold symmetry. And then here, looking down here, you have cyan, cyan, magenta, 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 and green. So here you have a 2-fold, basically relating one and the other by 180 degree flipping back and forth. Okay. So it's a beautiful structure. And so let me, forgot. Let me see whether I can do this. So this will take a little bit of time. So essentially, I'll show the surface of the structure. You can sort of see actually it's a very tight arrangement of all the protein molecules together. So there's really very little hole, very little cavity in the structure so that you can see. Because it's such a large structure, to render the surface takes some time. Even, well, it's on a laptop, so not that powerful. So that's best that can be done. All right. Okay, do you have any questions so far? Or this is too simple for you? It is? All right. Okay. Well, wrong coloring. There you go. So now you can probably see symmetry better, right? So here, the threefold right here, right? And then here you have fivefold, a little star. And then twofold is right here. Here's the twofold. And then I have to bring the mouse in. So this is the inside. You can see another color now, yellow. So that's the other subunit. So there are 4 subunits. And the fourth subunit is only on the inside. And it doesn't really have a domain. It's basically an extended structure. You could say IDR is intrinsically disordered protein, but it's a critical glue that bridges many, many interactions of the protein together to form this capsid. And then if I zoom out again, you can really see really no gap. The only gap you can find is right here. You can see a little hole right there at the fivefold axis, a little hole. Everything else is completely covered. So really remarkable evolutionary architecture to form something like this to encapsulate the viral genome. So I guess that's all I'll show you today. And then, so have you received the files from Professor Shukla? But anyway, you should have it. Do the exercise, and then tomorrow afternoon we'll do the first part of the session. Tomorrow afternoon we'll do the Q tutorial and the PyMol tutorial. But do some exercise at home first, so that you're familiar with it. So tomorrow we can go through them. Okay? All right. Thank you.